disarmed TI plasmids. We have discussed that TI plasmid, it is going to induce tumorous growth or crown gall formation in mainly dicotyledonous plants. So dicots are those plants, their seeds contain two cotyledons after germination. So that's why they are called as dicots. Now, this TI plasmid, which is natural or wild type, we can say that it is playing a role in pathogenesis, that it is going to infect the plant and resulting in tumor formation. Now, it is very easy to recognize that the natural TI plasmid, like other plasmids, it cannot be used as a cloning vector to transfer gene of interest to the plants. So, for this reason, some modified form of TI plasmids have been synthesized or constructed that lacks such ability, that is pathogenesis. So, if you look on the structure of the TI plasmid, it is actually the tDNA that is transferred to the plant, and it is doing each and everything that is in to induce the infection or tumorous growth. And all other genes on the TI plasmid, they are required for the transfer of the tDNA. Now, in modified or disarmed TI vector or plasmids, tDNA portion has been removed and it has been replaced with gene of interest or certain selectable markers. No tDNA no more there, and it has been replaced with our gene of interest. तो इसकी वजह से इसकी जो ability होती है कि वो ट्यूमर को इंड्यूस करे वो खत्म हो जाती है. तो इस तरह का जो वेक्टर होता है जिसमें ये ability लैक करती है कि वो ट्यूमर को इंड्यूस करे, we can call them as the disarmed TI plasmids, and they can be used as a cloning vector to transfer our gene of interest. So, in one example, tDNA has been replaced with some portion of the artificially constructed PBR322 plasmids. If you remember, we have discussed this plasmid many times previously. So, one portion of the PBR322, it is used to replace tDNA in the TI plasmid. Now, in addition to that, nopaline region of the tDNA, it has been maintained because this nopaline will act as a selectable marker. So we can study one of the example of the disarmed TI plasmid. This modified plasmid, it is called as PGV3850, and it is actually a derivative of PTIC58 plasmids. इस प्लाज्मिड में जो tDNA का पोर्शन है उसको रिप्लेस कर दिया गया है PBR322 के DNA से. इसमें एक एम्पिसिलिन रेजिस्टेंस की जीन भी मौजूद होती है, लेकिन कुछ पोर्शन जो है वो tDNA का, जिसमें नोपालाइन प्रोडक्शन या पॉइंट प्रोडक्शन की जो जीन होती है इसको मेंटेन रखा जाता है. जैसे कि मैंने बताया था कि tDNA के राइट पे या लेफ्ट पे जो हैं वो 25 बेस पे रिपीट्स होते हैं जिसके दरमियान tDNA होता है. तो इस जगह पे फॉरेन DNA को रिप्लेस कर दिया जाता है. और जो विर जीन्स हैं ये अपना नॉर्मल फंक्शन परफॉर्म करती हैं. पहले ये tDNA को ट्रांसफ़र करती थी, अब ये वही काम करेंगी फॉरेन जीन के लिए. तो फॉरेन जीन जो है वो नेक्स्ट प्लांट सेल में ट्रांसफ़र हो जाएंगे. नो आफ्टर द फॉरेन DNA has been inserted randomly at any site in the host genome, then the next question is that how the transformed cells they can be selected. As I told you that here a small portion of the tDNA it is retained, that can result in the synthesis of nopalin biosynthesis. So nopalin products, they can be screened. In addition to that, some other biomarkers that are related to herbicide resistance or drug resistance, they have also been used. The reason is that sometimes the screening of nopalin products, it is time consuming, or some enzymatic assays may have to use in order to detect the production of nopalin products or synthesis. So because of this reason, alternate selectable markers have been used, and those selectable markers, as I told you, that they may be based on drug resistance or herbicide resistance.